Yes, so all this is based on the soil food web method that was developed in the US by Dr. Elaine Ingham and is further being developed and refined and taught within her soil food web school. And actually, I have went through all the courses there, and I have recently, or I will soon become a soil food web consultant. So basically, what I would like to do at this moment is to kind of put a ground, like a base to what we are talking about here today, and what are we talking about when we are talking about the soil, because there are many different ways of thinking about soil, and the way I'm going to explain it now is kind of a paradigm shifting way of thinking about soil, which is different from the conventional thought, and that is that the soil is the vibrant ecosystem. And so it's a complex, and when it's healthy, it has the continuing capacity to function as a vibrant ecosystem. And it supports all life on Earth. Together with the plants, it creates a thriving and vibrant ecosystem. So when we are talking about the soil as being an ecosystem, it means that it is very, very complex, that it's far beyond our grasp of current understanding of what exactly is happening, but we are beginning to kind of scratch the surface, no pun intended, to understand what is actually going on and how the soil is one of the major elements on Earth that supports the whole life. So usually when we think about soil, we think about the mineral part of the soil, which is like mineral crystalline structures of the bedrock, or the clay particles, sand particles, and based on that we characterize our soils of being clay or silty or loamy or sandy, and so on. But these crystalline structures actually contain all the elements that plants need to be growing in a healthy and disease-resistant plants, but they are kind of locked because they are in stable crystalline structures within those particles. The other part of the soil, which we recently started to pay attention to, is the soil organic matter. And when we talk about that, we usually talk about soil organic matter being like wastes, residues of plants, animals, and microbes. And at the same time, it is a habitat and food source for those plants and microbes. And it's a really important link in the carbon cycle. So, and then we have like different structures of the soil organic matter that could be simple sugars and amino acids that could be readily available to be taken in by plants or microbes. But then we also have, with the help of the microbes, the organic matter over time becomes more and more complex until it's stable. It's stable until we disturb the soil, of course. But then the other way in which carbon enters the soil, which we kind of don't think about it, is the carbon that comes from the plants that are photosynthesizing. And it's so-called liquid carbon pathway, which is coined by Dr. Christine Jones. And this is a really fundamental way in which carbon actually enters the soil, because plants are the only ones that can convert the sun energy into the energy that can be used by all the other beings on Earth. And this is not to be dismissed. Plants have the capacity to, basically, they are the source of all food on Earth. And it is we also don't usually think about what is the plant's capacity to photosynthesize. But when the overall system is balanced, when it's working properly, then the plants can photosynthesize in their full capacity. And what we think conventionally is that plants only can photosynthesize about 15 to 20% of their innate genetical capacity. But of course, that highly depends on the whole system and how degraded or how thriving it is. So basically, what I'm trying to say here is that the plants are a part of the soil. They cannot not be. And the third part of the soil, the fun part for me, is the soil microorganisms. So when we're talking about the soil microorganisms, we're talking about bacteria, actinobacteria, fungi. These are the pictures of some beautiful pictures of fungal strands, then protozoa and the nematodes. And the soil actually contains, just in 1 gram, it contains several thousands of different species of microorganisms and 200 up to 200 meters of fungal hyphae. Of course, it depends of many different factors, and a billion bacterial cells. So they must have some kind of role in that soil. And I want to say that, of course, they do, and they have a fundamental role in the soil. It is calculated, I don't know how, but it's a fun fact, from 550 gigatons of all living biomass in the world, 450 gigatons are plants, but from the remaining 100 gigatons, 93% of the mass are microbes, bacteria, fungi, and protozoa. And all the other animals consist of only the rest of like the 7%, and humans account for only 0.01% of biomass, which is kind of funny. So definitely soil microbes have a really important role, not just in the soil, but also in every living being. This is where we started from a few billion years ago. So the soil food web works like this. The plants are photosynthesizing and they're exudating the excess of the photosynthase in their root zone because they want to lure, attract, and feed the microorganisms that are usually in bigger concentrations in the root zone because there is food. And so basically, primarily are the fungi and the bacteria are the ones that are attracted to the root system, and they are feeding on the plant exudates. It is oversimplified what I'm talking about now because it's, like I say, very, very complex thing. There is also something like something that is called rhizophagy, which means that plants can also take up bacteria and fungi and protozoa by themselves in the root zone, in the roots, and they can kind of synthesize those microbes and use them as a food source as well. But yes, so fungi and bacteria are the ones that are within the root zone. They're growing there, feeding on the root exudates, but then this activity also attracts nematodes and protozoa, which are the higher trophic level in the soil food web, and they're basically consuming the fungi and the bacteria. And what they excrete, what they poop out, is something that is plant available. So plants can use, it's like a micro manure from these microorganisms, protozoa and nematodes. So this is something that is happening every minute of every day. Then basically plants themselves create a feeding ground for themselves by the activity of photosynthesis. Then we also have the higher trophic levels like microarthropods, arthropods, other animals, insects and animals. But the basis of biodiversity is actually thriving and healthy soil microbiological content. And from there, all the other biodiversity is kind of built up upon. The plants are actually the ones that are leading this game. They exactly know which kind of exudates, which kind of sugars, which kind of sugar compounds or amino acids to excrete from the roots in order to attract the sets of microorganisms they need. Because there are many bacteria that could mine nutrients from the bedrock, from the soil mineral particles, and that's how they get to those minerals, because when the protozoa eat those bacteria or eat those fungi that are also really good miners for the minerals, this is what becomes available to the plants. And this is the base for the soil structure. The soil structure is balanced or built when there is high enough microbial activity within the soil. And as you can see in these two pictures, the picture on the left shows a well-structured soil, and the picture on the right shows a poorly structured soil without these aggregates that are visible. And when we look at those aggregates under the microscope, we can also see that the poorly structured soil is missing the aggregates. There are only mineral particles visible, while the well-structured soil has those aggregates in place. And so basically, soil also consists of air and water. So when the soil is very well structured, 50% of the soil is actually air and water, because this is what plants need. This is what soil microorganisms need. They're aerobic organisms that thrive on oxygen. So basically, the soil structure is something that is kind of a bank account for our growing operations, because if we don't have soil structure, then we don't have air, we don't have water, and that's the main problem within today's mainstream agriculture. Unfortunately, we don't have a lot of time to say what are all the benefits of the balanced soil food web, but actually everything that you can imagine that a healthy soil can provide, all those ecosystem services, is what I have listed here. So the soil structure and the nutrient cycling that is restored and is working all the time. And then when you have the plants that are photosynthesizing well, they can also use up, they can build upon this strength and be disease resistant. And the balanced soil food also supports biodiversity. So there are many benefits to that. One principle on which the soil food web method works on is the principle of the ecological succession and the soil food web. It's known to all of us that the ecological succession above ground, how it works. So we have bare soil, and then we have annual plants, perennial plants, vegetables, shrubs and bushes and vines, and then we have deciduous trees, forests, and old growth forests. So this is how it goes. If we would let our environment alone, then everything will become forest one day, probably much slower. But basically, what I want to say here now is that the above ground succession is actually followed by below ground succession. Like I said, the plants are leading the game. They are deciding which organisms are going to live, which organisms they want to feed. So when we are talking about the below ground succession, we are talking about bacterially dominated or fungally dominated soils. And it is expressed in the fungal to bacterial biomass ratio. So basically, when you don't have much bacteria, when you don't have much fungi, and the soil is bacterially dominated, it will benefit mostly weeds or early successional crops. When the fungal to bacterial ratio is balanced, then we can grow vegetables, cereals, perennial grasses, and fungally dominated systems are crucial for growing anything woody, like vines, bushes, orchards, forests, and so on. So when we want to know, we know what we want to grow and what kind of successional stage benefits that plant, but then we also know what is in the soil, and then we can compare and see what is missing actually. Of course, this is oversimplified as well, because we also know what kind of what kind of microorganisms we want to see. We want to see also the biodiversity of microorganisms there. We want to see the protozoa and nematodes in certain numbers and so on. Bioactive compost that we developed here at AhlmanEdu, or compost, well-made compost, is an excellent source of adding those microorganisms in the soil. And what we have done here in Alman, we are basically trying to make a good compost, which we are very successful at, I have to say. And then we are testing it in different environments to see how it works in these local contexts. And when we have, there is, of course, rules how to make a good compost. You know what kind of feedstock you want to put there, how to manage the compost pile, when to turn it, and things like that. But what we get in the end is a micro-rich soil amendment. We also started last year to make farm scale composting. So we have a compost turner, and we're now experimenting on that. But when we are talking about the bioactive compost and what does it mean that is good, we know what kind of minimal numbers of which microorganisms we want to see in that compost. And our bioactive compost has much more than minimal numbers. So it's a really high quality compost that we accomplished to make. And but besides the microorganisms that are coming with the compost, of course, you also get the other benefits. It's organic matter and it's the nutrients, but the nutrients are in so-called reduced form. It's not, it's very important that all the feedstock, all the materials that went into the compost, that are decomposed completely, because then you have those stable compounds like enzymes, vitamins, amino acids, organic acids, antioxidants, antibiotics that benefit soil and that benefit plants. And they can be, because they're stable, and active. They're stable and active. They also, through these like fine signals that with which microorganisms communicate, they are also activating the maybe dormant microorganisms within the soil that you're adding this compost to. We can use the compost as it is, but we are also adding compost in a form of compost extract or compost tea. So compost extract is basically mechanically extracted microorganisms from the compost, and the compost tea is something different. It is aerobically grown microorganisms from the compost in this like a tea brewer by adding food to the liquid and of course air. The compost extract is usually good for as a soil drench, so to basically to inoculate soil with the soil organisms, and the compost tea is usually used as a foliar spray because during the active phase of growing within the compost, the tea brewer, microorganisms, namely fungi and the bacteria, they are creating these glues, and so the compost tea can glue them itself to the plants' surfaces. Okay, so this is the base, and now I'm going to tell you about all the trials that we have done over the years in the on the Ahlman and elsewhere. So the first one was the perennial flower bed test. We have this perennial flower bed where we divided it into 3 different plots. The one was the plot that is going to be that was treated with the compost extract. The second plot was treated with conventional mineral fertilizers, and the third one was the control plot. And what we wanted to see is how the compost extract will affect this soil. And this is like typical soil that is used in landscaping. So it didn't have much of anything. I will show you now. But when I tested in the beginning, I was testing how many bacterial biomass there is, how much fungal biomass, and then also how much beneficial protozoa and how many nematodes there are in the gram of that soil. And we also know that those are perennial plants there. So basically, we also know what are the ranges that we want to accomplish by adding those compost extracts. So we have some base range that we could compare the results to. So the initial state of this, the initial state of the flower bed, the trial plot was that it was bacterially dominated, and it had some fungi, but not enough, and it had also some not so good root feeding nematodes, and very little protozoa, and the fungal to bacterial ratio was like there was practically no fungi there, and so the fungal to bacterial ratio was not in favor of the plants that were growing there. But then after 4 applications of compost extract, we managed to, by adding protozoa and nematodes that were eating all the bacteria, we managed to lower this number significantly, and then we also inoculated the soil with the with enough fungi that remained there, that lived. So the fungal to bacterial ratio became almost balanced, which is a very surprising result that we had. This was in year 2022, and last year I wanted to test what happened after a dormant part of the season, and what do I see in the last spring. So we still got enough beneficial protozoa and nematodes, and fungal numbers dropped a little bit, but the bacterial numbers still stayed the same. The last year's applications was just to put the fungally dominated compost. It's Johnson Sioux compost that acted as a weed barrier, as well as a source of nutrition and microorganisms. And this year we are going to test again and see what happens. But when I was comparing the results from the trial plot and seeing what was happening in the control plot or what was happening in the conventionally managed plot, when we are looking the fungal biomass, we can see that this very pale line here represents the conventionally managed plot. When we applied mineral fertilizers, the fungal numbers were actually sliding down for the first 3 compost extract applications, or the first 3 assessments, microbiological assessments, and then it started to kind of climb up. But this is the trial plot fungal biomass, which climbed significantly. And then also this is a comparison between the fungal and bacterial biomass ratio, how it stayed the same in the control and the minerally conventionally managed plot, and then this biologically managed plot, we managed to kind of make this fungal to bacterial ratio closer to the desired range, which is 2. Okay, this is another experiment. This is done in our vegetable garden last year as well. So we have these 2 plots for carrots. This is a permanent bench cultivation, and these 2 plots were actually divided into 3 different trial plots and 3 different control plots. And what we did is we have put like a thin layer of last year's compost, and then we treated the seeds with the bioactive extract. They were just soaked for a short period of time and dried, and then